In late apoptotic cells, now listen, with respect to flow cytometer, late apoptotic cells behaves like a necrotic cell. That means DNA will come out. That means in a late apoptotic cells, both phosphatidyl serine will be on the surface and DNA is coming out. That means the late apoptotic cells will be stained with both propydium iodide as well as FITC, annexin 5, conjugated FITC, right? That means suppose if I just okay, now listen with all your attention. When this first cell was interacting with the laser, what will happen? It is what? Early apoptotic or late apoptotic? Late apoptotic. That means here is the late apoptotic signal. Why Because it is showing both annexin 5 FITC signal as well as PI signal. But here, what is this? Late apoptotic or early apoptotic? This is what? Early apoptotic. Again here. This is what? Late apoptotic or early apoptotic? Late apoptotic. That means what? Here. Right? So now you know. Now you rest of the things where it will be placed. Here. Now listen. Suppose if I say that this is a cell and it is neither stained by propydium iodide nor it is stained by annexin 5 and conjugated FITC, what will happen to this? What do you mean? This means this is a viable cell. What is the definition of viable cell? It is completely healthy, that is, it's a living cell. Okay, so this is a viable cell. Now viable cell will give any red signal or green signal? No signal. That means it will be placed here. Clear? It will be placed here. Now, this is what? Late apoptotic cells. Late apoptotic cells. This is what? Early apoptotic cells. This is what? Necrotic cells. This is what? Viable cells. So, this is the basic of how we can analyze cell death or apoptosis, necrosis or apoptosis using flow cytometer. Now, in the next class, we will be discussing something called FACS, fluorescence assisted cell sorting. What do you mean by fluorescence assisted cell sorting? Listen, every flow cytometer is not a fax machine. FACS, it's fax. Every flow cytometer is not a fax machine. What is a fax machine? Listen, fax actually uses the principle of hydrodynamics and laser, okay, but it has got another unique property, that is, suppose this is double positive, suppose this is necrotic cell, it will throw the necrotic cell here, early apoptotic cell here, apoptotic cell here. That means it will sort out the cell, which cells are which, and they will differentiate between these cells, and they will separate these different types of cells in different types of counters. This is called FACS, fluorescence assisted cell sorting. Sorting means what? Suppose if I tell you that in this room, divide the students, okay, who are girls and boys. You will sort out, this is a boys section, this is girls section, right? So you are actually sorting it out. So this actually sorting means what? Categorizing a particular population. categorizing a particular population is called sorting. That means in a cell sorter, what will happen? Different types of cell will be sorted according to their surface markers, according to their different types of things which are according to the different types of markers, as well as according to their different type of shapes or something like that. Okay. So in the next class we will talk about in details the fax machine and how it works. Okay. Thank you.